Well, here we are live. I'm very happy to welcome you to this announcement of the Canada Gairdner Awards for 2023 and to welcome actually all of our awardees to the platform today. They're all sitting backstage and they're going to join me on stage to answer a few questions that I have for them, that some students have for them, and that if you have questions as well, you can put in the chat. I notice that already there are many congratulations in the chat, which we will pass on to the awardees. And please, if you have questions, put them there. I cannot guarantee, given time restrictions, that we'll get to all the questions, but we'll certainly pass them on if we don't. So what I'm going to do is to ask each group of laureates to come forward and address a few questions. So the first, I'm going to pull up Drs. Bassler, Greenberg, and Silverman to the stage. Hello, hello. Nice to see all three of you in person. I hope you enjoyed the video. It was kind of fun, and it really emphasized some of the aspects of your work over the years. And it's really great to have all three of you here to discuss your work on quorum sensing in bacteria, which has really been an evolving story over so many years. So I'm going to start with Mike Silverman. And Mike, you began working on this one little bioluminescent bacterium, Vibrio fischeri, and how that community signal leads to this collective glowing bioluminescence. Did you ever imagine that the field would evolve to where it is today?
No. If you can believe it, this story started many years ago. And for me, the genetic analysis of this phenomenon, it started 40 years ago when my graduate student, Joanne Engelbrecht, cloned the genes for bioluminescence from this bacterium that lives in the light organ of a squid. It's called Vibrio fischeri. And Joanne cloned the genes and analyzed them, and we came to the conclusion that Vibrio fischeri controls bioluminescence in this density-dependent manner by measuring the presence of itself. And that was very interesting, very peculiar, but was it exciting? Well, we got a kind of an ambivalent response to that work. But we were also working on another luminescent bacterium named Vibrio harvii, and it was superficially similar to Vibrio fischeri, and we were trying to isolate and understand the genes that control luminescence in that bacterium. But we were making any headway at all. We were really jammed on that until Bonnie Bassler came to join the lab. And she hadn't done a lot of genetics before, but she was very eager, very eager. Anyway, to make a long story short, and I'm really shortening this because Bonnie spent almost 4 years working on this problem, and the result was we found that Vibrio harvii was just a real gab about it. We wanted to talk to everybody. And Vibrio harvii was bilingual, and now Bonnie tells me that Harvey is actually multilingual. Anyway, that meant that this organism could sense molecules in its environment. We call it talking, but it's really chemical sensing, and it can sense all these various things in the environment and integrate the signals and modulate the signals and control the light coming out. And so this This was very interesting. This meant that there were many possibilities for getting conversations going, so to speak. And so that made us start to imagine um, what it might mean. And we started imagining that in these big bacterial communities that there's really it's not chaos. There's a lot of organization there, and this organization comes because there's a lot of communication going on. And so uh, uh, this became much more interesting, and people started looking at their own research and wondering whether maybe this is relevant to their work. And I guess you could say it's the area of research started to explode, and
Well, I think obviously, Mike, it's still exploding today, and it's wonderful to see from that beginnings where it's at today. And you can look back proudly on the beginning, and you can look back proudly on recruiting Bonnie to take it forward. So let me switch to you, Peter, and let's jump forward all those 40 years to where the field is today. And you know, with the Gairdner Awards, we like to think about the impact on human health and disease. And obviously, there was a bit in the video about biofilms and how, you know, different kinds of infections are related to the fact that bacteria talk together and form communities. How do you see the field moving forward in terms of really trying to treat diseases? What can we learn from quorum sensing?
Yeah, so that's a great question, Janet. We're all interested now. We know that there are lots of different bacterial species that cause disease, and they use quorum sensing to coordinate themselves uh, and be effective at causing diseases. You know, the big problem with antibiotics is that resistance emerges, and there are theoretical reasons to imagine why resistance to a quorum sensing inhibitor will be difficult, because we now understand that quorum sensing is a social activity. So with a traditional antibiotic, imagine it stops bacteria from growing or it kills bacteria. And if a single cell emerges that is blind to that inhibitor, it can grow and it will take over. But with a quorum sensing inhibitor, we're blocking the ability of the bacteria to work together as a community, maybe in a biofilm. And if a single cell emerges that is blind to the inhibitor, it won't be able to grow because it requires the cooperation of all the members of the community. So this is all theoretical and has sort of come from our increased understanding of how quorum sensing is coordinating social activities. So, you know, looking forward, I think there's a lot of potential to develop therapeutics that target quorum sensing. We don't quite know how to do it yet. So we need two parallel tracks. People that are interested in developing these chemicals are important and understanding how they work, but we still need to know more fundamental biology. And the two tracks have to go hand in hand. And I have to say, the part that I'm good at, if I'm good at anything, is the fundamental part. So that's where we're pushing.
Great. I think obviously in science in general and in the kind of awards we give, the fundamental science is what drives everything else. Without the fundamental science, we're not going to see the translation. So it's an important message. So Bonnie, I had my question to you, which was to sort of go beyond bacteria and how bacteria talk to other cells, including the cells of our body. So the interaction between the microbiome and our body, and how do you see that? Where's that field going? It seems to be a very exciting one right now. I know it's hard to predict the future, and you only have a couple of minutes to do it. So tell us what you think about this field.
Sure. Yeah, thanks, Janet. Right, so we now know that we have this magical consortium of bacteria called the microbiome that keeps us healthy, and it works hand in hand with our human immune system to fend off predators, invading bacteria, and quorum sensing is playing an increasing role in that. And so we're really interested in wondering whether we can exploit that knowledge to make pro-quorum sensing molecules. So could we instead of fighting against the pathogens, like we've always done in medicine, could we actually make pro-quorum sensing molecules to make the chit-chat that's going on in our microbiome even better at the expense of invaders? And then we also now, you alluded to this, we also now know that this communication extends beyond kingdom boundaries. So human cells are participating in these chemical conversations with our bacteria. And so my gang is also wondering whether we could boost the human part of these chemical conversations at the expense of the invaders. So we're really trying to exploit not just the pathogens, but the beneficial bacteria and our own biology as well to make new kinds of medicines. And of course, it all comes, as Mike started this, from this quirky glow-in-the-dark bacterium and learning that these bacteria could communicate, make groups, and get more bang from their buck by acting in collectives. And so now we'd like to exploit and mess up those collectives.
That's great. So it is a great story from the single bacterium to things, the interactions we're talking about today. Somebody's got a dog. I'm going to thank all three of you, but don't go away. I'm going to bring you back in a little while. And now I'd like to bring Dr. Hassabis and Jumper to join me. Good morning, good afternoon, wherever you are, and welcome and congratulations again. It's great to see both of you in person, and have a couple of questions for you. I'm sure there were probably lots of questions in the chat to both of you, but Demis, to start with, your career trajectory, you've talked about this in many other circumstances, but leaving DeepMind, you began with chess, you worked on video gaming, you did a PhD in neuroscience. You've said that all this, which seems somewhat random, has actually been a necessary route to your success in artificial intelligence. Tell us a bit more about how you see all those things fitting together.
Sure. It's a pleasure to be here. So, they actually, if you look back on my career, there is a lot of different things that I've done, as you say, but the common thread actually is I've always been obsessed with artificial intelligence and the potential of it to benefit society since I can remember, really, since I was a child. And so all these things I've picked to do have been different skills or different ideas that I thought could eventually help with artificial intelligence research. Just so starting with games and specifically chess, that's what got me thinking about the nature of thinking, and also my first contact with AI was using chess computers, old-fashioned ones where you actually had to press the squares down on the chess computer, and that made me think about how do we program, how are these machines programmed to actually be able to play chess. I thought that was kind of magical in a way, and that led me on to studying computer science and working on artificial intelligence in the games industry. And then I did a PhD in neuroscience in order to also learn from the brain, like how does the brain support certain functions for intelligence, things like memory and imagination. And then so all of those different aspects then fed into the beginning, the founding ideas for DeepMind. And then, of course, we use games again, maybe most famously with our program AlphaGo, to first program to beat the world champion at the very complex game of Go. And we used games as a training ground to test our AI algorithms on. It's a very efficient way of testing our AI algorithms. But really, the ultimate goal was always to generate general purpose algorithms, somewhat inspired by neuroscience, and then apply them to really important real world problems, specifically in science. And that led us to AlphaFold and trying to solve the problem of protein folding.
Yeah, oh, it's a wonderful career path. I think it's interesting for the students out there to realize that you did have an idea about where you might end up, but you took a long, circuitous route perhaps to get there. So it's great. So John, we talked earlier, actually, your background is also perhaps not what you expect for a computer science nerd, but you did condensed matter physics, theoretical chemistry, which is all sort of relevant to AlphaFold and protein structure. But obviously for DeepMind and leading the team for AlphaFold, you had to build an interdisciplinary team. So how do you do that? How do you really bring together the sets of people? Because obviously we're recognizing the two of you, but I fully understand that there's a team, big team behind AlphaFold, and you're really here to represent them. So how do you build a team?
Absolutely, yeah. And I was just reflecting after Demis's answer that I kind of took the opposite path. That I bounced around whatever looked important or interesting or useful, and ended up in a very fortunate place. But I think also kind of fortunate to be at DeepMind, where it's really made of a lot of people that are very passionate about solving these problems and solving these problems however we can, and especially when we're thinking about science problems and AlphaFold in particular, the problem is so very important. We went after it any way we could. And we knew really from the beginning that this was a hard problem, that lots of really smart people had worked on this for a long time, 50 years. And so we were going to need new ML ideas if we were going to have a chance to solve this. And we put together a team that really reflected that, with a ton of top tier ML researchers and maybe one or two people with protein experience, including myself. And so it was really a team that was interdisciplinary and weighted toward like where we hoped or thought we would have to go, or what we brought distinctively to this problem. But kind of from my background and from everything else, I'm really convinced that the most creative ideas don't just come from one person knowing proteins and one person knowing ML, but really having lots of people that know everything. And I think what's been one of the most exciting and rewarding parts of the project has been watching everyone learn and grow from each other, and kind of ending up with a whole team of structural biology, ML experts over the course of it. And I think that's a lot of the kind of creativity and what we found in this hard problem of how do you kind of marry proteins and ML. One of the reasons we were successful, I think, is that we built a team that was thinking about these problems in a really interesting, sophisticated, and new way. And I think that's really, really important, that ML for science, like there's been lots of ML for vision and text and lots of great general ideas, but putting them together in these really hard scientific problems, I think, requires specific and creative thinking. And I think we were able to build that team over time, and because everyone in it was so very committed to the importance of this project.
That's great, and obviously building those teams sets you up for the future and new initiatives. So you have the team ready to go off in new directions. It's fantastic. So thanks to both of you. I'm going to put you backstage, but no, I'm not. I'm going to keep you on stage. But first of all, we're going to show. We've sent before the announcement. We asked some students for general questions that they would like to ask the awardees. So we're going to run a video now from Sarah Kidd, who has a student question for you, and I'll bring back the quorum sensing team as well in a minute. So let's hear from Sarah.
Hello, I'm Sarah Kidd, and I'm a grade 12 student who's interested in the field of biomedical science. I'd like to first commend you for your integral contributions to biomedical science research and tremendous accomplishment. Congratulations. Obviously, no amazing advancements come without challenges. So with that in mind, I would like to ask, what were some challenges that you encountered during your research, and how did you overcome them?
Okay, so now we have everybody back, and I'm not sure I'm going to be able to ask everybody to answer this question, but perhaps I will start with you, Bonnie.
Okay, sure. I think when at the early days, one of the big challenges was to get this idea that bacteria could communicate, to get traction, right? People thought that being able to have group behaviors and communication, that was the purview of higher organisms. And of course, the only organism that we had that we knew did it was an obscure bioluminescent bacterium. So it was really challenging to get people to believe that this happened and that the ideas and principles we were finding were general. But scientists do what scientists do. We all just kept making more data, more bacteria were discovered that we had quorum sensing, and then scientists like data, and so the idea percolated, and that is how we overcame the challenge. It was just by keeping going.
Okay, so how about John, challenge that you had to get in AlphaFold?
I think one of the really big challenges was we really knew, like from the beginning, there were two hard problems in protein structure. At least kind of coming from my background, I see it as two really, really hard problems. One is what kind of Leventhal made famous, Leventhal's paradox, that there's a huge combinatorial space of how a protein might form a 3D structure. And so we kind of thought, okay, maybe search is hard. How do we understand that? And then we also knew, though, that protein energetics are subtle and really poorly understood. Like, what is this an answer? Like, if you show someone a structure, they can't say yes or no immediately. And so how do you understand these things? And it was really, I think, unclear throughout the project. Would we really focus on the search and the optimization? Would we focus on recognizing solutions? Do we have to do it kind of very much like molecular dynamics? There were a lot of different kind of paths and forks. And I think one of the really incredible things was to watch this kind of slowly collapse. As whoever started to make progress, we would pile into different areas. And so we were always exploring, but try and contain that exploration as well. And so I think really finding that fine balance between different kind of views of the problem and then working together with the team and using that all into one system was very difficult and very exciting as it worked. And I think the answer ended up in a place that none of us really expected in the beginning or in the middle, but it was kind of proven out and developed over time.
So Demis, more on a sort of career level, on a more personal level, challenges that you've had along the way?
Well, there's been many. If you look at now how incredible the AI field has gone, when we started DeepMind back in 2010, almost no one was working in AI. It was almost a field that's sort of, very difficult to raise any funding for, and almost no one was working in. So it was sort of pretty brave sort of for us to think that some progress could be made. And I remember when we were working on our early systems, and we were trying to use relatively simple games like Atari games from the 70s, things like Pong and Space Invaders, really simple games where we were trying to build systems that could learn for themselves just from the pixels on the screen. And I remember we were struggling for a couple of years to get these systems to do anything. And maybe, we were thinking maybe we were a decade or two too early. And sometimes happens in science. But we persevered with it, and then we made a few early breakthroughs, and it's been a kind of roller coaster ever since then. But it's been amazing to see, be sort of in the vanguard of seeing how AI has flourished and really kind of renaissance of the techniques in the last decade to where we are now. And for us, our proudest moment is with AlphaFold.
So maybe a decade or two too soon. So how about in the quorum sensing? So Peter, were you a decade or two too soon? Were there challenges along the way there?
So I think being a decade or too soon put us in position to be at the center of this whole thing. If I were to have answered the question that Bonnie answered, I would put it exactly the same way. We, for 20 years, there was a small group studying these funny luminescent bacteria, and no one else was interested. But we were in position when information started coming in from people who worked on other bacteria that told us that they had these same kinds of systems. So it was sort of fun. For a while, Mike's lab and my lab were the only labs in the world working on this, and we were the only people interested in each other's work. And that all changed 20 years later. And so I don't think we were too soon. We were, you could say we were pioneers. It was great fun if you're willing to have nobody else interested.
That's great. That's great. Mike, did you have just one point you wanted to add very briefly, or have we covered the background here?
Well, why do people want to focus on talking together or communication. Why is that sort of the holy grail of research? And it's because recognition and sensory processes are at the top of the pyramid of information flow. And if you can start at the top of the pyramid and understand that, understand these communication networks, you can work down through the hierarchy and discover all the genes and the proteins and the functions that are regulated by them. And so it's strategically, it's a wonderful place to be. It's up at the control center. And I think that will be the key to understanding really complex problems like communities of bacteria. They look chaotic. They look like a pile of mud, but or some other substance. But there's a lot of organization and communication and structure there, and it's important for us to understand that world because it does interact with the macro world too.
Yeah. So for students, you have to take an interesting question, you have to stick with it, you have to explore all different ways of carrying the research forward, and then you too can be successful like the people we have on stage today. So thanks to all of you, and now I'm going to ask Dr. Belizan to join me. Hello.
Good morning.
Jose. Good morning.
Nice
How
to
are
be here.
you? Nice
Fine,
to
fine,
see
thank
you.
you.
It's very nice to
Very
see you
pleased and
indeed.
grateful about this distinction.
And congratulations again.
Thank you, thank you.
So I have a sort of general question for you on your work. So you really worked with vulnerable populations in South America. That's where your work has focused. It's led to evidence-based interventions, improving maternal health in pregnancy and childbirth.
Yes.
And I guess my question was, how did you make sure that those findings became international recommendations? How did you ensure that they had global impact?
Yes, I would say one thing, that those living and working in the Big South, we have an advantage and a set because we live in the same environment of people who want to serve and focus our research on their needs. So being in a person-to-person control, working in hospitals, working in small villages, working in slum areas, we can really know what the people need, what are their priorities, what are their needs. So we focus on research on their needs because that's the question that we raise, saying that okay, that the people who like to improve their quality of life, the satisfaction, also the health action, also some say also about survival. So let me give you a small example. When I was a medical doctor, every women coming to their antenatal control during pregnancy, they told me about their fear about episiotomy. They say, okay, doctor, episiotomy is so hard. I hear from many women about this. And then we say, well, we are doing episiotomy to every woman, and they're not evidence to do it. Then we did a big trial, very well designed, with a lot of power, and we demonstrate that episiotomy does not signify any benefit, no, including harm. And then, well, okay, we published this in the Lancet, and then the international organization took this, like WHO and providers. And we're very proud that this study, with this question raised by the concern of the people, signify that nobody now is doing a routine episiotomy in the world. So that's the way we really react. And I say it's an asset to live in the same environment and to really receive what people think there are priorities, and also that serve us to better implement in these people. And we have a lot of rapport with people. Social science for us is very important. Before starting any study, we speak with the populations, setting how they feel, what they want. And that's why our studies are very, very proud and pleased that really are converted in actions for the global at global level, no, and focusing in equities too, no, to achieve equity in what we provide.
So I think that's a great answer, and I think the fact that you communicate with the communities that are affected is really important.
Yes,
And I think we're
yes,
going to hear the same message when we talk to Dr. Musquash with his work with Indigenous populations in Canada.
yes,
This
yes,
ability and the necessity to really ask,
yes,
well, what do the communities want and what do they need? So that's a
yes.
great lesson. I wanted to ask you a sort of more general, sort of more personal question. So, how did you get into this? When you were starting your career, who were your inspirations?
You know, in Latin America happened something curious and rewarding. When I was resident, we heard from physiologists and researchers very close from here in Montevideo in Uruguay that he's doing research and he conceived the term perinatology, that is around birth, and he raised the relevance of the fetus. You know, when I was a student, there are a lot in obstetrics books about women care and women, but not too much about the fetus. And then he wanted to start to say, it's important because we can do wrong things if we don't care the fetus and regard only maternal health. So I went there, I said, doctor, I want to stay here with you. And then he said, okay, come back. I can't pay you anything, but we realized with my wife to go there. And then after that, it came, okay, that's where I received the training. But my training was always in the South Cone, in Argentina, Uruguay, Chile, where I do my PhD. So there are here potential in our countries, but I insist we need to really be very powerful on research methodology and use very careful and research methodology.
So that's great. So you really did get your inspiration, and you've developed your whole career in South America, but it's obviously had impact worldwide. So that's great.
Yes.
So I'm going to ask you to hold on in the back room. I'll be back, bringing you back to answer a student question in a minute. So congratulations again. And now I'm going to turn to the first of our Momentum awardees, Dr. Christopher Musquash. Good morning. Hello, Chris. How are you?
Morning. I'm doing well. Yeah,
Good.
likewise.
Congratulations again.
Thank you.
So I think there is a sort of connection between what we just heard from Dr. Bellosan and your kind of research as well, because really, building on your expertise as a psychologist and your many community-based partnerships and collaborations, you've really developed evidence-based practices that impact on Indigenous mental health and substance abuse, but they align with First Nation values. So tell me the importance of cultural-based knowledge and community input in your work and how you integrate that.
Sure. I think that, there's recognition that many of our communities are really challenged with difficulties in mental health and substance use. But at the same time, there's, tremendous knowledge and wisdom that communities have in terms of understanding the nature of those challenges, the nature of the difficulties, how it was that those challenges came to be. But importantly, what's needed for healing and how to go about doing that. So I think, similar to Dr. Bellosan, ensuring that our work is really driven by community priorities ensures that we're better able to address the needs of those communities in a manner that's responsive to the realities of the context that we find ourselves in at present. So this really, allows then to incorporate a lot of culture-based knowledge that's grounded in identity and in that way, using that wisdom to develop solutions. And this is really the pathway to healing that our elders and people and communities have been advocating for a long time.
So when you were building your career, how did you really move to ensure that you maintained your connections with the community as you developed a more sort of Western style approaches? How do you integrate the two, and who were your inspirations along the way, I guess?
Yeah, I see myself as someone who's just been very, very fortunate to have been able to meet and learn from so many mentors, both in psychology and academia, but importantly from community, as well as cultural people and elders. And so, for me, I see the way in which the psychological approaches and culture approaches working together is really ensuring that there are people that can kind of translate, in a sense, between the underlying constructs, let's say, that we're trying to understand and trying to support. So I often joke that I sort of accidentally ended up in a place where I was able to develop a whole bunch of opportunities to try and help, and really it was through mentorship and just people who supported that. I think I really am really fortunate.
And again, the same sort of question I was asking Jose, which is how do you ensure that the approaches that you've developed in Indigenous communities, obviously there's a lot of lessons to be learned from mental health interventions across all communities. So how do you personally work to make those linkages happen?
Yeah, I think, contributing to any and all conversations that you can, and really finding a place for traditional knowledge and culture-based knowledge, in broader conversations. It's through advocacy. It's through recognizing that, people, although different, coming from different places with different, backgrounds and traditions and so on, ultimately, are very interconnected with the natural world, with history, with the contemporary context, as well as the future. And so, the scientific research associated with psychology and psychopathology or intervention or whatever is really now becoming far more interdisciplinary and expanding well beyond the individual difference variable to understand interrelatedness among biological systems and social systems and environmental systems and so on. And I always have been hearing these conversations from our cultural people and our elders for a long time. That knowledge is very, very old, and we are able now to enter into conversations where there's more understanding across those different ways of knowing.
That's great. So that's a wonderful story and a wonderful connection for us all to think about, for sure. Okay, congratulations. Don't go far. We're going to come back in a minute to address a student question. And now I'd like to ask Gairdner to join me. Good morning, Gairdner. How are you?
Good morning. Good, thank you.
Very nice to see you, and congratulations again. So
Thank you.
I have to say, we've talked about this before, but you are still a rare breed. It should not be, but you are a rare breed, a female neurosurgeon, and a very successful one. And I'm sure that students out there are wondering, how did you get where you were today? So what convinced you that this was the right chosen path, and who were your sort of supporters and mentors along the way?
Great, thank you very much. And Dr. Rossent, if I may just take a very quick moment to thank you, the adjudicators, the Gairdner Foundation, for this once-in-a-career opportunity. It's really an amazing recognition of the work I do. And so, why neurosurgery? The organ, our brain, is such an intricate and complex organ that it really fascinates me. It continues to amaze me that it makes us who we are uniquely in many ways. And neurosurgery in particular requires significant dexterity, delicate movements, focus, concentration. And through that, we really try for each person to do the best that we can to preserve brain function. And it's really extremely rewarding when we have good outcomes for our patients. And so that continues to really motivate me to do what I do. It's that focus and concentration that I really enjoy in the dexterity that it brings. And then on top of it, of course, to understand why disease processes happen, it's a intellectually very stimulating field to be in. We understand the brain very little, and so any research that we can do to understand the disease processes that it impacts will advance our field. So for those reasons, I really feel quite fortunate and privileged to be able to have this opportunity on a daily basis to do what I do.
And so your research is trying to classify brain tumor subtypes by non-invasive methods. And your research is one of those areas looking at non-invasive detection of cancer, which is a growing field and a growing interest. How do you see the future impact of non-invasive testing in brain tumors, but also more broadly?
Yeah, great question. And of course, it's my group and many others in the field that are trying to make that advance to better understand the biology of tumors, what drives malignant progression, and most importantly, how we can in fact personalize therapeutics and treatments. I think the biggest impact, if we use the large data sets that we've accumulated, hopefully using AI that we just heard about earlier, is to get to a place where we can come up with predictors of who's at risk of having brain tumors. Because unfortunately, by the time that the most malignant brain tumor presents to us as surgeons and healthcare providers, it's a bit too late. We need that one single test that could tell us who's at risk and be able to intervene earlier. And it's through the biomarkers that we're investigating, using the large data sets that we have, that would be able to give us that predictor of who's at risk. And more importantly, equally after surgery, after treatments that we implement that are standard, what are the biomarkers that are going to determine response to therapy, and to then be really able to individualize, personalized response to treatment for any one person with brain tumors.
That's great. I think it is a very exciting time, and it must be exciting to be at the edge, as you say, linking your clinical practice with the research that's going to impact on patients. So thank you. Congratulations. Don't go far. We're going to hear a student question, and I'm going to bring you, Dr. Musquash and Dr. Belezan, back to answer the student question.
Thank you.
All right. Hello, everyone. My name is Charlotte Domingos. I am currently in my 4th year of completing a public health science degree. And I would like to say congratulations, first off, for winning this award. You must be very, very proud. As an award-winning scientist, my question is, do you have any advice for young, aspiring scientists out there?
So I'm sure that's a question that we all get asked, and I'm sure the rest of our awardees would also have advice on this one too. But I'm going to ask Jose for your advice to young scientists.
Yes, one thing, we need young scientists. I don't know why you think different. Your neurons are different. So yes, we need you. Please join us. Very well, one. And we say that it's a passionate career. It's a passionate career. Whenever you see that you can involve better health, better quality of life, that's the most gratifying thing that person, a scientist, a doctor, or whatever provider can feel in their life. So I'm sure if you start this career and do it with passion, please, passion, you will feel very, very free when you see what you have done.
Thank you. Passion is a word I use a lot as well.
Yes.
I suspect, Gallery, what's your advice?
Yeah, actually, I think, first of all, thank you for the question. It is to find what you're most passionate about. And it's not easy. It doesn't come immediately. But once you find that, I think the key for me would be that ultimately who we are as individuals, what our convictions and principles are, defines where we end up. and how successful we are. And it's not the single events that define our career paths or our impact. It's the collective and the totality of everything that we do. But I think ultimately your principles and convictions really provide that motivation to drive you forward and define your long game of your career.
And Chris.
Yeah, thanks. It's a great question. I'd say pursue things even if you're not sure how it's going to turn out, and then persist and find mentors who share your values and who are willing to encourage and support in all those times where you may not be certain as to where you're going.
So those are great messages from all three awardees. And as I say, I think all the awardees and myself would share those sentiments. For future scientists, I hope that you've enjoyed the interactions we've had today. Thank you very much, Dr. Belisan, Zade and Musquash. And thank you, the audience, for staying with us, having a chance to hear me chat, and you join in talking to our inspiring awardees today. I hope you've enjoyed it. I just want to say again, congratulations to all our laureates. And please, all of you, go to the Gairdner website, gairdner.org, and follow us. We will be having more events throughout the year. And in October, we have the Gairdner Science Week, where all of our awardees will be back in Canada to travel across the country, and you'll get a chance to hear from them again. This particular presentation is being recorded, and it will be on our website very soon. So well done to everybody. Congratulations, and enjoy the rest of your day.